I really like.
When in my head I was like that, and then I was like, because then she just kind of started like trying to give her cheese. Okay, I'll bring her in. So I started out. Oh yeah. Yeah, that's fine.
Oh yeah, I checked.
Oh, whatever.
So
The right is out.
so much.
Oh yeah, we are.
Like, what are you doing? He's like, oh, good job.
Versace's like, yeah, I'm so ready.
I think I didn't much. I wonder if like you need that.
It's better than.
Amino acids. Okay, so that is sort of your background material for learning about each amino acid, which takes some time. We're continuing on today, talking about the formation of proteins and peptide bonds, but I also want to point out another aspect about some of the amino acids that we will need to learn, and that is related to the pKa. You can focus on this table, mostly on this part of the table, in terms of the pKa of some of the amino acids that have the ability to ionize, meaning they can protonate and deprotonate. They act as acids or bases, right? And so what does the pKa mean? You remember? Yeah.
50% or 50% are basic, like deprotonated.
Yes. So the pKa is the pH at which a group, an acid, is 50% protonated and 50% deprotonated. So what this means is there are 7 amino acids which have the ability on their side chains to protonate or deprotonate. So that means they can become neutral or positive charge or what have you, depending on what their side chain looks like, if you change the pH above or below their pKa. And we're going to do some work in the problem set on exploring that idea. So the lecture is pretty short, but I'm going to probably get at during the problem set and kind of go through some of the questions on that so we can understand. Okay, lots of other numbers here. This is also useful. We're starting to help you memorize the 1 and 3 letter amino acid codes. If you have individual amino acids, meaning amino acid just by itself, there are 2 other groups on amino acids that can protonate or deprotonate. And that is the carboxylic acid end of an amino acid. You do not have to memorize each of these individually. We will say that the pKa of a carboxylic acid is around 2, okay, for the carboxylic acid end. The amino end of an amino acid is pretty variable. So we'll say that's about 9, around there, okay? And then 7 of our amino acids have the ability to protonate or deprotonate on their side chain. So not all of them do. Okay. This is a pretty useful overall. Sorry. This one is pretty useful overall for coalescing all the amino acids in one. Yes.
Since the pKa of the ionizing side chain is more variable, do we need to memorize the 7 of those?
Those aren't so variable because those are for each individual amino acid. So each individual amino acid, 7 of them, depending on if it's an acidic amino acid or a basic amino acid, tyrosine, and also cysteine. So those have specific pKa's you should know. This image here shows what the amino acids look like at pH 7. Okay, so this is their charged state at physiological pH. if you start to, so that becomes important to sort of know what these side chains look like. When I say you don't have to be able to draw the amino acids, but you should have a pretty good visual image of what these side chains look like. Aspartic acid and glutamic acid, if you look at the table, their pKa's are around pH 4. So at pH 7, they are deprotonated and they have a negative charge. Okay. This is because we are above the pKa of these side chains. We're also above the pKa of the carboxylic acids. That's why those both have a negative charge. Those side chains have already lost their protons. Okay, so that's why they're pictured in this way. They're being pictured at pH 7 because of physiological pH. Okay. We'll get into that a little bit more in the problem set too. All right, so you should know these pKa's because each of these side chains have an individual pKa that is specific to that amino acid. That being said, memorizing those numbers for those individual amino acids. When you have an amino acid in the context of a whole protein, its pK of its side chain might be different than the published number. And so we will see some examples of that in certain enzymes. So just understand that this is a typical pK range. I do want you to know these numbers though, but it can change in the context of a protein. So what does that mean? So it might mean that if you have a lysine, which is a basic amino acid, it might mean that if it's next to other charged groups, it's going to be less likely to deprotonate. And so its pKa will be higher than you would expect it to be. Because when a lysine deprotonates, it becomes neutrally charged. And maybe if it's near other nearby charged groups, it doesn't want to deprotonate. So its pK is a little bit different. So we'll talk about specific cases. All right. So you have a lot of information about individual amino acids. So definitely start learning those so that doesn't creep up on you and be something you're cramming for before the test. But you'll want to spend your time studying other stuff. Okay, so how do we make a protein between two amino acids? The interaction is going to be between the carboxylic acid end of one amino acid and the amino end of the other. Those two interact and dehydration synthesis occurs. There's a water lost in that reaction. So one oxygen here, two hydrogens there, and we lose a water and we form a peptide bond is what that's called. What enzyme actually does this in the cell? What catalyzes the peptide bond? How do we make protein? Yes, ribosomes, yes. Ribosomes are actually catalyzing this. This is not happening on its own, right? So in the context of a ribosome, the amino acids come in on the end of tRNAs and then they're put together by the ribosome. Okay. So we now have a dipeptide there. We have two amino acids linked together by a peptide. And this is a longer polypeptide chain. We have 5 amino acids linked together. So much like the directionality of a nucleic acid, when we state a sequence, we state it from the 5' to the 3' end when we're talking about DNA or RNA. We talk about a directionality of proteins too. So by convention, we always state the amino acid side chain that is on the amino terminal end of a protein. Here's the amino terminal end. This is tyrosine, and then we write the sequence going towards the carboxylic acid end. of a polypeptide sequence. The ends of these are the only parts of a protein. Once you start making a protein like this, the ends have the charges, the amino charge and the carboxylic acid. The other backbone groups where we've made the peptide bonds here, 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 those have lost those charges because we've lost the hydrogens and the oxygen that were in individual acids charged. When you make a peptide bond, those are gone. So only the amino end and the carboxylic acid end have charges here, except for if you have side chains that are charged. And that will depend on what your overall protein composition is. For this polypeptide, what is its overall charge? Yeah, it's 0. It's net neutral. We have a +1 charge and a -1 charge. None of the side chains here have charges on them. And that would be different if we had aspartic acid, lysine in there. We'll talk about that in a bit too, in the problem set. Questions on this? Okay, so when a peptide bond is made, this is the true peptide bond. This is between the carbon that was part of the carboxylic acid and the nitrogen that was part of the amino group. That's the peptide bond. But the electrons can exist in a conformation where the carbon is double bound to the oxygen, or they can temporarily be in this conformation. I'm going to go back and forth between these two. And so because of that, the peptide bond has what's called a partial double bond characteristic. Because there's this resonance structure between these two. The electrons kind of go back and forth. So people have measured the length of the peptide bond. A single carbon-nitrogen peptide bond would have this length, and a double bond would have this length. A peptide bond is in between, so it is truly like a partial double. The important part of that in terms of a protein and how its atoms move around each other is that a partial double bond cannot turn. So the carbon and nitrogen relative to each other cannot rotate. They're held in place, and you get something like this between the regions of 2 amino acids. So this is the peptide bond. These two atoms cannot turn relative to each other. And so the carbon nitrogen, this also would have partial double bond characteristic. And all of these atoms, these 6 atoms, are in a plane relative to each other. Okay? So they're all on the same plane. They're planar. That is not to say that all of these atoms are. So this bond and also this bond can rotate. So this group could twist. This one still stays in the same plane. And we'll talk about the rotation, that these are called the phi and the psi angles. And those can rotate. And that's important to making a protein that isn't just a sheet. Our proteins fold up into all of these structures. So there is rotation. It's just not, these 6 are in a plane relative to each other. And then you can imagine another 6 would be in a different plane and so on. So when a peptide bond is made in the context of a ribosome, you've attached 2 amino acids together. This is the peptide bond. Once you make that, remember those atoms don't turn relative to each other. So the directionality of the 2 amino acids across that will remain in place. So when this bond is made, you can imagine that the side chain, which is shown here in green, attached to the alpha carbon, same here. This peptide bond is permanent with regard to its rotation. So the side chains, when this bond is made, could be on opposite sides of the peptide bond, shown here and here, or they can be on the same side of the peptide bond, shown here. So this is what's called a trans conformation, opposite of the peptide bond, opposite sides, and this is a cis conformation. Which one do you think exists more frequently? And why? Yeah.
The trans is lower energy, and so there's less steric hindrance versus the cis.
Yeah, so there's less steric hindrance. And what we mean by that is that the side chains are less likely to try to be occupying the same space. The atoms here are very close together. Depending on how big the side chain is, they might even be trying to occupy the same space. So energetically, this is much more favorable conformation to be in. So typically see side chains in the trans conformation relative to each other. And you'll see, so if you have just a linear polypeptide chain shown like this, you will see the side chains on opposite sides of the peptide bond. Here, here, and here. Okay. See that most frequent. Questions on that? Could we get a twist in the protein so that these two become cis conformation? No, we can't, because this bond doesn't rotate. So once it is sort of established in this trans conformation, it will stay that way. Yeah.
That's because like this resonance structure makes it possible for it to be a pi bond, right? So it's kind of like
Yes. So the resonance structure is such that they you can't get rotation around this. So that we're not getting a movement and flipping over of that bond. Okay. So I'm gonna let you get started on the problem set. Well, let me do one first. Let me do